Did you know that when a medical AI system hallucinates a chemical structure,
Mm.
it might not actually be glitching?
Right. It might just be using those hallucinations as like a molecular scratch pad, just literally doodling its way to the right answer.
Which is wild. I mean, we are going to unpack that mind-bending concept today. Welcome to this deep dive. Our mission here is incredibly ambitious. We are tackling a massive stack of 20 cutting-edge research papers from the bleeding edge of medical AI.
It is a dense stack of research, but it reveals a fascinating transition.
Yeah, and we are committing to giving you a tight audio overview of all of it, right within a 15 to 17-minute window. Whether you are a doctor, a tech developer, or just someone curious about who or what will be diagnosing you in the next few years, this is your ultimate shortcut.
Exactly. Because we are moving past the phase where AI just identifies patterns and entering this era where it actively reasons, adapts to chaos, and, well, even designs new biology.
Right. So to keep this massive amount of information organized for you as we race the clock, we have one ground rule. We will strictly mention the paper number right before we dive into its specific findings. That way you always know exactly where we are on the map.
I like that rule. Let's start with the foundation. Because before we can trust these systems with human lives, we have to understand how they actually absorb and organize medical knowledge.
Which brings us to paper 11. This research looks at medical foundation models, and it drops a pretty counterintuitive finding.
It really does.
When these massive neural networks learn, they converge on a shared understanding of medical images. But that understanding is driven entirely by self-supervision, not by the clinical labels that humans provide.
Yeah.
Like, how does an AI learn about a disease if we aren't explicitly pointing to it and saying, hey, this is pneumonia?
Well, it learns through reconstruction. In self-supervised learning, you take an image, say a chest X-ray, and you digitally masked out chunks of it.
You just hide the data.
Exactly, you hide it. Then you force the AI to predict what should be in those blank spaces based on the surrounding context. By doing this millions of times across millions of unlabeled images, the model is forced to learn the underlying physics and geometry of the human body.
So it's like a kid learning the rules of physics just by playing with blocks in the yard, rather than, reading a textbook. The underlying structure of the data teaches them more than our human labels do.
Yes, that's a great analogy. And what paper 11 proves is that our human labels, our textbook definitions of diseases, actually do less to align the internal logic of these models than just letting them observe the raw, unlabeled anatomy.
Wow.
The AI develops its own internal representation of health and disease based on structure, not vocabulary.
But if it's building its own internal geometry, how do we know what's actually happening inside its brain? Paper 10 tackles this head-on.
This one is staggering.
The researchers figured out how to actually peek inside these frozen 3D CT vision encoders. And they discovered that specific clinical findings are encoded in a remarkably sparse set of just about 10 vision encoder channels.
Just think about the scale of that for a second. You have a model with hundreds of millions, sometimes billions of parameters. It's a vast ocean of mathematical weights.
And out of that entire ocean, a specific type of lung abnormality is controlled by just 10 little levers.
Right. The mechanism is highly localized. It's akin to looking at a massive audio mixing board for a stadium concert and finding out that out of 1,000 sliders, just 3 of them completely control the lead singer's microphone.
That's crazy.
Paper 10 shows that if you artificially turn off those specific 10 channels, the AI's ability to spot that one specific lung finding completely collapses. Meanwhile, its ability to see everything else remains perfectly stable.
That level of mapping is wild.
Yeah.
It means these systems aren't just impenetrable black boxes anymore. We are literally finding the anatomical light switches inside the neural network.
Exactly.
And we are seeing this self-supervised approach scale up massively with paper 19. This study introduces BrainNext, which is a self-supervised foundation model trained on over 60,000 completely unlabeled brain MRIs.
And scaling to MRIs is a massive leap because MRIs are volumetric. You aren't just looking at a flat 2D photograph, you are navigating complex 3D spatial data.
Right, it's got depth.
BrainNext proves that self-learning without human labels is highly effective at identifying complex, irregular structures in 3D space, like finding meningiomas, simply because it has learned what a normal 3D brain is supposed to look like.
But there is an elephant in the room with all these massive foundation models, right? They require supercomputers to run. I mean, a local community clinic does not have a server farm in the basement.
No, definitely not.
That's where paper 20 steps in, shifting over to pathology. They highlight how models like GigaPath-Flash and GigaTIME-Flash take these massive computationally heavy models and just compress them.
Using a process called knowledge distillation, which is fascinating, you essentially take the massive unoptimized model, the teacher, and use its outputs to train a much smaller, highly efficient student model.
Oh, okay.
The student doesn't need to learn from scratch. It just mimics the expert intuition of the teacher.
And the results in paper 20 are phenomenal. I mean, GigaPath-Flash retains 97% of the billion-parameter teacher model's performance, but uses 50 times less compute. That makes it actually usable in real-world clinics.
We are finally bridging the gap between theoretical lab research and practical medicine, but the front lines are incredibly messy.
Which perfectly transitions us into the clinical side of things. Because while these models are great in a lab, I have to bring up paper 5 with a dose of skepticism.
Oh, path-agent-bench.
Yeah, paper 5 introduces path-agent-bench to test how well AI can find evidence on gigapixel whole flight images. Even though models can reason well, they are terrible at actually finding the evidence here. The AI's hit rate drops to a dismal 0.02 at high magnification.
Right. It's the soda straw effect. Imagine trying to navigate a massive map of a city, but you are only allowed to look through a tiny soda straw.
That sounds impossible.
It is. When the AI is zoomed in at high magnification, it can see individual cells with incredible clarity, but it completely loses its spatial context. It doesn't know where those cells are located within the broader tissue structure.
So it lacks that human pathologist's intuition to just zoom in and out to build a mental map.
Exactly. But AI is actually overcoming this real-world noise. Let's look at paper 1. This study introduces MSSA, which stands for Memory-Supported Synergistic Adaptation. It allows models to adapt to new medical images on the fly without forgetting their pre-trained knowledge.
Wait, how does it do that mid-diagnosis?
It uses an online memory bank. Normally, an AI's knowledge is frozen after training. MSSA allows the AI to make an initial pass over a messy image, identify features it's confident about, and store those in temporary memory. It uses its own predictions as an anchor to guide itself through the murky parts.
It's actively bootstrapping its own context. And paper 1 notes this boosts segmentation performance by up to 12.2%.
Huge improvement.
But what if you don't even have enough real-world images to train the model on initially? Paper 13 brings us AstraNet, which tackles a lack of clinical data for drug-induced sleep endoscopy.
Because getting clean camera data inside a collapsing human airway is incredibly difficult.
Right. So AstraNet trains on virtual CT scans to map out real human airways. It's like having a pilot log hours in a flight simulator before touching a real plane.
That's a perfect way to put it. And that adaptability is moving straight into the operating room, too. Take paper 2, which introduces 𝗪𝗵𝗁. Here, vision language models are being fine-tuned to recognize microscopic instrument-tissue interactions during robotic surgery.
So helping the surgical robot actually understand what it's cutting.
Exactly. Using a latent action-guided framework, it forces the AI's attention solely onto the tiny region where the scalpel or needle is physically interacting with the flesh. It forces the AI to understand the action, not just the picture.
That's incredible. But what about when AI doctors are talking directly to patients? Paper 8 introduces 𝗪𝗫𝗫𝗶𝗴𝗵𝗹. This explains a new way to evaluate medical chatbots by strictly decoupling their ability to collect a patient's history from their ability to spit out a diagnosis.
This is so important. Historically, we've graded chatbots purely on their final answer. But a doctor's job isn't a multiple-choice test. It's an investigation.
Yeah, a chatbot might be empathetic and gather a flawless history, but have terrible diagnostic logic. Or it could ask awful questions and just get lucky with a guess.
Right. By separating those two things, paper 8 brings much-needed scientific rigor to how we audit these chatbots.
Okay, so diagnosing, operating, talking, that's the clinical front lines. Let's shift gears to molecular biology and designing drugs, because this is where things get surreal.
My favorite part.
Paper 17 introduces 3D Fit, pointing out that while large language models are great at text, they really struggle with 3D spatial constraints when trying to design binding molecules. They lag way behind diffusion models.
Well, biology doesn't work sequentially like text does. It works dimensionally. A drug molecule has to physically fold and fit into a 3D protein pocket. Text-based logic just fails to account for physical collisions in 3D space.
Which brings us back to that wild teaser from the start of the deep dive. In paper 4, we see that when AI models reason through chemistry using a chemical chain of thought, they fabricate structural claims, they hallucinate constantly.
Yes, and it's a functional strategy.
Wait, really? So when the AI lies about a chemical structure, it's not actually failing, You're saying those hallucinations are structurally load-bearing, like the AI is just scribbling on a molecular scratch pad to figure out the real answer.
That is exactly what it's doing. Paper 4 shows that the AI lacks visual imagination, so it generates these physically impossible chemical substructures just to hold spatial data in its working memory while it solves the rest of the molecule.
Wow. If you force it to stop hallucinating, does it fail? Yeah, its ability to find the correct final answer actually drops. But obviously, when we synthesize real drugs, we cannot tolerate intermediate errors. We need physical precision, which is where paper 7 comes in with 𝗵𝗽𝗆. It explains how we bypass LLM limitations by using advanced structure predictors like 𝗵𝗽𝗁 to guide the design of high-affinity small molecules.
𝗵𝗽𝗁 acts as the physics engine. The LLM suggests the sequence, and the structure predictor immediately simulates how it would fold in real 3D space.
So it molds the AI's ideas to ensure they actually fit.
Exactly.
But we still need a lie detector for bad predictions. Paper 6 mentions reliable B. This evidential framework acts as a lie detector for AI drug predictions by calculating consensus among an ensemble of models.
Right. If the models fundamentally disagree with each other, the framework mathematically flags the prediction as highly uncertain.
And by filtering out those low-confidence protein-ligand pairs, Paper 6 shows it reduces prediction errors by 25%.
Which is critical when designing massive proteins like antibodies. We see this in Paper 3 and Paper 18. Antibodies are notoriously hard to engineer because they flex and bend.
Paper 3 uses ABOPD to solve this. They explain how AI is being used to design highly complex, flexible antibodies using privileged native geometry to keep 3D loops stable. Privileged native geometry sounds intense.
It just means teaching the AI the real-world physical rules of how a protein naturally rests and moves in a biological environment, rather than treating it like a rigid statue.
And Paper 18 pairs perfectly with that. It introduces AAMFM, an antigen-specific multimodal foundation model that maps these specific antibody-antigen interactions in a shared latent space.
The power to design these complex biological molecules is incredible, but, and we have to be honest here, it opens the door to terrifying security risks.
Yeah, let's take a serious turn here and introduce Paper 9. This is the bio-red teaming paper, Intern Biobreaker. This highlights a terrifying reality. Frontier LLMs can be jailbroken to generate modified viral sequences.
And they bypass the standard text safeguards by asking the model to interact purely in raw genetic sequences, those ACTG base pairs.
And in lab tests, these AI-designed pathogens were physically synthesized and actually showed enhanced infection potential. That is horrifying.
It underscores the absolute urgency of genomic surveillance. Which brings us to Paper 14, biosec bench surveillance. When we use AI agents to conduct genomic surveillance and spot these outbreaks, the results are really concerning.
Yeah, Paper 14 says the best models, like Opus 4.8, only get it right about 50% of the time. Why is it so bad at surveillance?
Because it struggles with choosing the right biological filters and e-value thresholds. It gets overwhelmed by the background noise of nature and can't always distinguish a benign mutation from an engineered threat.
Okay, let's breathe a sigh of relief and shift from global pandemics to personal health. Paper 16 introduces GlucoTune, which standardizes blood glucose data for diabetes without leaking sensitive patient info.
It anonymizes the data while preserving the core metabolic trends, which is huge for privacy. And speaking of privacy, Paper 12 introduces HiMe.
Yes. Paper 12 is fascinating. It's a platform that allows you to run a privacy-first, real-time health agent directly on your local device using your own wearable data.
Edge computing. The model is compressed enough to live locally on your phone or smartwatch, so your highly sensitive heart rate and sleep data never gets beamed up to a corporate cloud.
The math literally happens in your pocket. To wrap up our sources, let's look at Paper 15. This introduces MedGame, noting how AI is turning dry medical education into engaging storytelling games.
Right, it synthesizes clinical storylines for students to play through. The AI acts as a dynamic story director, altering the patient's vitals on the fly based on the student's choices.
It turns memorization into real problem solving. Well, looking at the time check, I can confirm we have successfully unpacked all 20 of these papers, and we are bringing this audio overview to a close right within our promised 15 to 17-minute window.
We did it. And it's been a phenomenal journey. AI is no longer just a digital filing cabinet for health data. It is a dynamic, sometimes hallucinating, but highly capable partner touching every layer of healthcare. From the smartwatch on your wrist, to the molecular structure of your medicine, to the operating table.
Before we sign off, what is the final thought you want to leave the listener with today?
Well, I want to connect back to our early discussion on self-supervision. If AI models develop their own internal geometry of health and disease, purely through self-supervision independent of human clinical labels, what happens when the AI's understanding of the body diverges from ours?
Oh, wow.
Could these models soon start diagnosing us with patterns of disease that humans don't even have a name for yet?
A completely alien understanding of our own biology. That is a brilliant and slightly haunting thought to end on. Thank you for joining us for this deep dive. Stay curious, keep asking questions, and we'll catch you next time.